speaker for today, and that's David Diemert. David Diemert is not only, I'll give you his bio, but he's one of those people that if you do what I do, you have a handful of what I like to call good citizens. David's a chair of one of our IRBs. He's on our IACUC. He's well-funded. He hasn't learned how to say no yet, and I hope that he doesn't learn how to say no because we would be lost without him. So David is a professor in the departments of medicine and microbiology, immunology, and tropical medicine at GW. Before coming to GW, he worked for 4 years at the malaria vaccine development branch of the US NIAID, where he was responsible for conducting clinical trials of novel malaria vaccines in the US and Mali. David earned his medical degree from the University of Alberta and completed internal medicine residency and ID and medical microbiology fellowship training at McGill University in Montreal. His research is focused on conducting clinical trials in the US, Brazil, and Africa to develop new vaccines, particularly for neglected tropical diseases. He has served as the PI on multiple clinical trials of hookworm, schistosomiasis, and malaria vaccines, and more recently, those for Zika, HIV, and COVID-19. In Brazil, David has worked in partnership with the Fundação Oswaldo Cruz, and I'm sure I butchered that, David, to develop a clinical trial site in a rural area of Brazil. More recently, he's developed a controlled infection model for human hookworm at GW to test the efficacy of the investigational vaccines and is serving as the site PI at GW for the phase 3 trial of the Moderna COVID-19 vaccine and the phase 2 trial of the Sanofi-Pasteur COVID-19 vaccine. Welcome, David. I'm going to turn it over to you.
Thanks so much, Sheila, for that introduction. I'm very happy to be here. Is someone going to be putting up the slides, or should I just share my screen?
That is up to you. Hillary, are you? Oh, there we go.
Oh, there we go. Next, please. So I'm going to spend today by quickly, because I know we don't have a lot of time, but I'll try to keep it to 30 minutes. So I'll leave some time for questions about our experience here at GW in helping to develop some of the COVID vaccines, one of which now is authorized by FDA. So I don't really have any disclosures, except that we're all very tired on the team here because we've been working quite hard on the clinical trials for COVID vaccines. But next, we're also very relieved and hopeful because we now have we have 3 vaccines that have been authorized by FDA, if you want to go next. And that just shows them there, the Pfizer, Moderna, and J&J vaccines, just in the order that they were authorized by the FDA. Next slide. And since then, since the first vaccine and in the middle of December got authorization, being the Pfizer vaccine, and then the following week, the Moderna vaccine, there's been a steady rollout of vaccinations. I'm sure we're all aware now we've reached more than 120 million people in this country, so almost 50% have received, of adults at least, have received at least 1 dose of one of the authorized vaccines. There's been a little blip lately because of the hold being put on the J&J vaccine. I'm not going to really get into that because of a lack of time, unless someone wants to ask a question about that. Next slide. But that's just really the tip of the iceberg. This is just from the WHO website a couple of days ago. There are still 91 different vaccines that are in clinical trials throughout the world. And there's more than double that are in preclinical development, so haven't made it quite to the clinical testing stage. You can't really, I don't have a pointer, but if you look down to the lower left, there's a whole list of the different types of technologies that are being used to develop vaccines. If you want to go to the next slide, please. But regardless really of the technology and the way that the vaccines are being made, they really have the same endpoint, and that is to develop neutralizing antibodies to this protein that you can see here on the surface of the virus. So that's the spike protein I'm sure we're all familiar with now. But really all of the vaccines, regardless of the kind of mechanism that they're working, all end up at the same endpoint, and that's developing antibodies or hoping to develop antibodies that will interfere with this interaction between the spike protein on the surface of the virus and the ACE2 receptors that are on the surface of our cells in the respiratory tract. And that's what the virus uses to get into our cells, into the respiratory cells lining the respiratory tract. Next slide. Sorry. And I just wanted to point out that there are two different kind of forms or shapes, conformations of that spike protein. So after it binds to the receptor, the ACE2 receptor actually changes form. That helps it to get into the cell. And that is actually important for vaccine development, because almost all of the vaccines that have been developed are based on that stabilized form of the protein before it binds to the ACE2 receptor. Next slide. So there's lots of different strategies that have been followed to develop vaccines. We're most familiar, I would say, with the ones on the right side of the screen, starting with the nucleic acid one. So there's both DNA vaccines and messenger RNA vaccines, the first ones that have made it to market, at least under authorization. There's viral vector vaccine, and the J&J Johnson vaccine would fit into there, as well as the AstraZeneca one. That's using a different kind of virus to carry the nucleic acid that encodes for the spike protein. And then the more traditional on the far right, which is the protein-based vaccines, recombinant protein-based vaccines like the Sanofi vaccine or the Novavax vaccine. There actually are a few groups in the world that are developing pure viral SARS-CoV-2 virus vaccines that are inactivated. So they just grow up the virus in the lab and inactivate it and use that. None of the companies in the US are really focusing on that kind of technology. There's some in India and China that are doing that. Next slide. I'm sure you've heard the term warp speed in reference to this effort. It's kind of changed the names with the new administration. But this is the list of the US government-supported COVID vaccine candidates that have at least been supported for clinical trials and or manufacture. There's, I think, 7 on the list. And you can see that kind of one of the strategies that was taken by the federal government was to put a lot of money into vaccine development, but also to fund a whole slew of different potential candidates using different technologies, because we didn't know at the start what was going to work. So this is one of the reasons I give for why we were able to develop a vaccine so quickly, is because there were a lot of different ones that were funded. And some of them haven't worked out, like the Merck vaccine has been abandoned, but we didn't know that at the start that it wasn't going to pan out. And luckily, some of the ones that were funded have worked and worked quite well. Next slide. The other thing that has enabled us to move quickly is kind of a paradigm shift in how we do clinical trials. So up at the top, you see the traditional paradigm, which is conducting the different phases of clinical trials in sequence. So from phase 1 through phase 3 before applying for licensure. That's really been compressed. And the way it's been compressed is not by cutting out any of the steps, but by overlapping them. So you can see down on the bottom, I'll show you an example of the Moderna vaccine that's been developed. The phase 2 was started shortly after the phase 1 vaccinations were complete and the first safety analysis was done in that trial. And the same thing happened with moving to the phase 3. So they were really done in parallel, but staggered, but allowed us to move a lot more quickly. Next slide. So this was the first vaccine that went into clinical trials. This was the Moderna mRNA vaccine. You can see one of the first people there in this press release getting vaccinated. This was started in Seattle, originally enrolling healthy adults between the ages of 18 to 55, and then subsequent to that enrolled older people. Next slide. And next. So this is the concept of the mRNA vaccines for those who aren't already familiar. So messenger RNA is a form of nucleic acid that kind of serves as the template for making proteins in cells. So the idea behind this is you take the messenger RNA that codes for that spike protein, stick it into these little fat particles called lipid nanoparticles or LNPs. And though that fat particle actually protects the RNA from getting degraded once it gets injected into the cell, into the body, and then also enables it to get into host cells, so into our human muscle and other cells after it gets injected into the muscle. And then those are our cells, the host cells, will utilize the usual protein-making machinery, ribosomes and so on, to make the protein, the spike protein from the virus. And then that gets exposed to our immune system, antibodies are made to it, and those are protective when you're exposed to the virus in the environment. Next slide. So this is from the Moderna website, just showing the timeline. So it went really quick from identifying the sequence, the kind of DNA or RNA sequence of the virus in January by the Chinese. They published that in middle of January. And this is another reason it was very easy to make a vaccine quickly, is because this technology, the mRNA technology, although there has not been a licensed vaccine using this technology yet, a fully licensed vaccine before this, there had been several that had been tested in clinical trials, so the technology was there to quickly make, as soon as you know the sequence, to kind of stick it into the sequencer, manufacture the mRNA, and then start doing the testing. So that could happen really quickly with this kind of technology. And then going all the way from mid-March, when the first person was injected in the phase 1, to the first person being injected in the phase 3 at the end of July. So really within 6 months. Next slide. This just shows some antibody results from that very first phase 1 trial that was done initially in Seattle, just showing they tested 3 different doses of the vaccine, giving 2 injections 4 weeks apart, and showing really good antibody levels after vaccination compared to people who had recovered from the actual infection that you can see on the far right of each of those 2 figures. And those, when you mix those antibodies that had been induced in people who got the vaccine with the actual live virus in the lab, it actually neutralized it. So based on these results, next slide. Oh, sorry, and I wanted to say too, this is really recent data too. Looking at that same phase 1 trial that was started about a year ago, but looking 6 months out after the second injection, and this is really encouraging, I think, in that the antibody levels really seem to be sustained over time, just going at 6 months after the second dose. People are always asking me, well, are we going to have to get booster shots? We're going to have to get vaccinated every year. I'm like, well, we don't know yet, because we don't know what we don't know. It's only been a year since the very first person ever received this vaccine. We don't know how long the antibodies are going to last. But the data so far are encouraging that they hold up over time. Next slide. So this is the network that was put together by mainly by NIH, kind of drawing on the existing clinical trial networks. GW has been a part of the HIV prevention trial network. So that's one of the reasons we were chosen as a site for the Moderna phase 3 trial. But this really network of sites was put together, sites that are used to doing clinical trials and vaccine trials, to do a series of phase 3 trials of different candidates. So the original goal was to enroll 5 different phase 3 trials, each enrolling at least 30,000 participants by the middle of this year. That actually was exceeded and happened much quicker than that. And really to harmonize the trial design, the endpoints, and also have a common data and safety monitoring board that would look at the results of all of the trials kind of in an independent manner, and also have a central network of labs that would do all of the antibody and other immune analyses. Next slide. Do you actually want to just go? Yeah, so it's harder when I'm not controlling. So this was the different trials that have been done so far with the mRNA vaccine from Moderna. So I mentioned that initial phase 1, all the vaccinations were complete, and initial data, safety data, and immunogenicity data was available by mid-May, which enabled the start of the phase 2. Same thing there, it enrolled really quickly, and that's another reason that we were able to move quickly, is because the trials enrolled extremely fast because we had so many willing participants. And then starting the initial vaccinations in the phase 3 trial for Moderna at the end of July. Next slide. So the design of the phase 3 trial was a traditional, placebo-controlled trial, testing 1 dose of the vaccine at 100 micrograms, 2 injections a month apart, and the endpoint was symptomatic infection. So you had to to meet the endpoint, you had to have a positive PCR test for the virus, and have symptoms as well. So we weren't tracking asymptomatic infection in this. Next slide. So this is the design again, and go next. So initially it was designed that we would do there would be two interim analyses done after 35% of the target cases and that symptomatic infection were reached. Then again at 70%, next. And that was based really on what was assumed was going to be the rate of these endpoints happening based on the incidence rate in last July. So that was grossly underestimated. It was estimated, the stats were that we'd have to detect at least 151 cases of symptomatic infection to be able to do the analysis. Next slide. Initially was estimated that that would happen in July of this year, that we would reach those 151 cases, but it actually happened much quicker because cases went crazy in the US and incidents went up. So we actually, by middle of November, we'd gotten almost 200 cases were recorded in the trial in study participants. Next slide. So this is who was enrolled overall. This is from the New England paper that was published at the end of December. Just over 30,000 people enrolled. We enrolled we just missed 350 at GW. In about 10 weeks, we enrolled that number. Next and next. So just some things to point out is in overall, about a quarter of the study population were over the age of 65. 20% were Latino or Hispanic, and then next, and just over 10% were African American. That was very important for us to try to get a diverse population in the study to really reflect the population as a whole, but also because we know that people of color have been disproportionately affected by this pandemic. And we did much better, if you will, at GW. At our site, we had about a third of our population over the age of 65, and over 50% were people of color in our study population. Next slide. So this enabled, so as I mentioned, just under 200 cases of symptomatic infection recorded in the study participants. Of those, 185 were in the placebo group, so resulting in a vaccine efficacy of 94%, or just over 94%. And even more encouragingly, although there were 30 severe cases of COVID and 1 death due to COVID, all of those were in the placebo arm, so 100% efficacy against severe disease. And this efficacy really held up across the different subgroups that were analyzed. So all of that allowed the application for the emergency use authorization at the end of November, which was granted just before Christmas, or on the 18th of December. Next slide. Next, so these were published in the New England Journal of Medicine. Next. This is the typical incidence, so it's a really nice visual to see how most of the cases were in the placebo arm versus the vaccine group. Next slide. And just to point out that regardless of age, sex, race and ethnicity, and having risk factors for severe disease, this vaccine efficacy really held up across all of the subgroups. Next slide. That's not to say it's not without side effects, and these are kind of expected, but in some cases they were quite severe. You probably, I'm sure many people on the call have received one of the Pfizer or Moderna vaccines. They're pretty much similar in terms of side effects. So pain and soreness at the injection site, and then some people had systemic or kind of generalized symptoms, headache, fatigue, sore muscles and joints. In some cases, to the point where people said they couldn't get out of bed for a day. But they were these are short-lived, don't usually last more than 24 to 36 hours, and no one had any significant complications due to these. Next slide. There were some rare adverse reactions that had been recorded in the phase 3 trial. So there were some allergic reactions, 3 cases of anaphylaxis in the trial in the 30,000 people. Those were all determined or assessed to be unrelated to vaccination because they happened more than 10 days after vaccination. We know now with millions of people getting this vaccine that there is a very small rate of severe allergic reactions, but it's not much more than you would see with the influenza vaccine. There were some cases of Bell palsy that were reported to the FDA. The FDA actually thought those weren't more than a background rate. And since now that it's, millions of people have been getting, it doesn't seem that this has panned out to be something related to vaccination. We had one of these cases of people who had had dermal filler reactions, or dermal filler injections for cosmetic reasons, and had a swelling of the lips, cheeks, under the eyes where they had people had received these injections, even if that occurred months, even sometimes over a year earlier. And this also has turned out to be a rare side effect of the vaccine. Next slide. There are still some unanswered questions. We're getting more data now, and actually CDC just published data on pregnancy, getting people or women getting the vaccine during pregnancy. These pregnant women were not enrolled in the trial, but some did get pregnant during the trial. And CDC now has come out with data that say that basically there's no adverse consequences of getting vaccinated during pregnancy after they studied people post-vaccination. There are pediatric trials underway. This vaccine, the Moderna vaccine at least, is only licensed for over the age of 18, although there is data now on down to the age of 12 years with both the Moderna and Pfizer vaccines. And more data is coming online showing that these vaccines, the mRNA vaccines, actually seem to be blocking asymptomatic infection as well as symptomatic infection. Next slide. So what happened after all of that? The EUA came out just in the middle of December. All of our volunteers wanted to know if they got placebo or not, and they wanted to get the vaccine if they did. So we started unblinding after a protocol modification was approved by the IRB right after Christmas, I think. And we started unblinding on the 30th of December. And we just wrapped up our final, we call them decision visits, where people come in to get unblinded on the middle of March. So we've unblinded all of our volunteers and given the vaccine to everyone who got placebo. Next slide. So that was our experience with the mRNA trial, the Moderna trial, and just one slide to show very similar results we're seeing with the Pfizer mRNA vaccine, which is very similar type of design and platform. So again, 95% efficacy at preventing symptomatic infection. Next slide. And I just wanted to say a few words of about some of the other vaccines, because I still think I have a bit of time. This has been the AstraZeneca vaccine, probably has been more people in the world have received this vaccine than any other, although it is not yet licensed or are under EUA in the US, and they haven't even applied for it yet. There have been a number of clinical trials that have been done. This is a viral vectored vaccine, so quite different technology than the mRNA vaccines. So what they've done, this was originally developed at the University of Oxford. They've been working for years on developing vaccines that use this chimpanzee adenovirus vector into which the gene encoding for the spike protein has been inserted. This is a non-replicating adenovirus. And then it's alive, but it doesn't replicate once it's injected into the body. So a number of clinical trials have been done, including a phase 3 trial funded by the US government through the Coronavirus Prevention Trials Network. That included sites in both the US and overseas. Next slide. This, I just said, this is the strategy, so that using that chimp virus into that carries the gene for the spike protein. Next slide. And you can hit next, I think, 2 more times. Yeah, and then this just shows the original efficacy that was published. This is not including the trial that had sites in the US. This was, I think, just in the UK, Brazil. I think just the UK and Brazil. And it showed an efficacy of about 70% at preventing COVID of all severities. But again, it was 100% effective in preventing severe disease and hospitalizations, which is what the main thing that we would like to prevent. Next slide. And there was some odd results from this data, though, in that some people, it turned out they got a lower dose of the vaccine than others. There was differences in the schedule of vaccinations. Some people got their second dose much later, weeks later than other people in the trial. if people who got the lower dose seem to have better efficacy. There is a lot of controversy about these results and how they did their study design, but it seems like efficacy between 60 and 70% is what has held up over time. Next slide. And this is also a 2-injection strategy. So these were just recent that came out about the phase 3 trial that had sites in the US, just recently published. This is showing about 79% efficacy at preventing symptomatic infection, again 100% against severe or critical disease and hospitalization, and similar efficacy even in older age group. Next slide. So the next. So what's next? I just wanted to finish off with a few other updates. So the J&J Janssen vaccine is also a viral vector vaccine, also an adenovirus vector, although this is a human adenovirus. That's the vector as opposed to a chimpanzee one. This, as I mentioned, the emergency use authorization came out at the end of February. This has been paused because of rare potential side effects of these clotting disorders associated with low platelets. The FDA, or the ACIP, the advisory group to the CDC, is actually meeting, I think right now, to go over the data again, and we'll hear later today or tomorrow maybe if that hold will be lifted. Next, the Sanofi vaccine. I'll just finish off actually our experience with this trial. So phase 2 testing of this was started in February and is ongoing. And then I mentioned the Merck vaccine is not going to move forward, and that actually development of that vaccine has been abandoned. Next slide. So this actually was just published a few days ago in New England Journal of Medicine. This is the Janssen J&J vaccine, so showing efficacy next of 66%. This is a single dose, single injection. So after a single injection of this vaccine, 66% efficacy at preventing symptomatic COVID, but better efficacy at preventing severe disease. So after doing all that work on the Moderna trial, and I should say it's still ongoing. We just finished we're still in the midst of finishing the 6-month follow-up visits. We're going to follow our participants for another 1.5 years to see how long the efficacy and the immune response lasts. We took on a second trial, and that's the Sanofi phase 2 trial. Next slide. This actually went into the first clinical trial starting in September. And this is probably more typical of what we see when we're doing clinical trials. So I think another reason we were able to move really quickly in getting licensed or authorized vaccines is because we had some luck in that we didn't have to go back and reformulate or remanufacture or figure out a better way to manufacture with the mRNA vaccines. But that has happened with, for example, a more traditional vaccine like a recombinant protein vaccine mixed with an adjuvant, the Sanofi vaccine. So this initial phase 1 didn't have great immune response in older people. So the company went back and changed the vaccine dose, the formulation. Next slide. And that went into another phase 1 trial, and then subsequently a phase 2 trial that we're a site for. So the trial that we're doing now started vaccinating at the last week in February, actually fully enrolled within the space of under 2 weeks. We enrolled 44 of the 720 participants. That was done at about 25 sites throughout the US and Latin America, testing 3 different doses. Thank goodness they removed the placebo because I'm sure we wouldn't have been able to enroll people if they knew they might be getting placebo. One of 3 different doses, they were randomized to 2 injections 3 weeks apart, intramuscular injection, just like the other vaccines. Next slide. This is just the study design. The main endpoint is actually 2 weeks after that second dose of the vaccine. So we've actually already reached that with all of our volunteers. And they're going to use the antibody data from that time point to decide on the dose to go through into a phase 3 trial. It's almost definite that the US, there will not be sites in the US for doing a phase 3 trial because it's going to be a placebo-controlled trial, and it would be next to impossible to enroll in such a study here. Next slide. So I think I made good time, and there will be time for questions if anyone has any. These are just some photos of our team. That's me in the lower right with Dr. Malkin, one of the other investigators on the study, the pharmacy, study coordinators above and in the clinic. We're doing most of the visits at the MFA, but we are using a trailer, as you can see in the bottom there, that was lent to us by the Department of Defense that we're also using to see study participants to kind of expand our capacity, because we had enrolled so many people in the study. And people generally prefer going to that because they can just drive up there and don't have to go into the clinic, which potentially in a pandemic could expose them to COVID. Next slide. So it was a huge effort. It has been a huge effort that continues, lots of people involved. And it's a real great collaboration we've had between School of Medicine, the Medical Faculty Associates, and also the School of Public Health at GW. Dr. Magnus on the upper left there leads the team at the School of Public Health that it's been great to collaborate with to try to work on recruitment strategies, data management, and the whole shebang. So it's been a lot of people that I'm representing today. And with that, I think I'll end and take any questions if there's time.
Thank you, Dr. Diemert. That was a very interesting presentation. It was great to have a firsthand account of the vaccine clinical trial process, and seeing that timeline really emphasized how quickly the vaccines were developed and put into use. So we have some time now, so we'll get into some questions. The first question is, can you please comment on the current Moderna vaccine trial for infants and children?
Sure. Hi, Mariel. So we're not involved in that trial. So I just know what I know from reading about it and hearing from the company we have weekly calls with them. So I know it started, and and it's a staggered trial. So it's enrolling first kids between the ages of 12 and 18, and then moving kind of in a stepwise fashion to younger kids, but it's going down all the way to 6 months of age, infants 6 months of age. As far as I'm aware, it has not reached the youngest cohort yet. So I know that they're fully enrolled for the 12 to 16 months. And I think there's about 3,000 kids that are going to be enrolled. I'm still not completely sure if they're going to base back to the FDA for authorization just on immune response. But I know they are following for cases of COVID, both symptomatic and asymptomatic. But I don't know much more than that, and when they'll be going to FDA for authorization. I would assume that, especially for the younger kids, it wouldn't be until the fall.
Okay, great. And the results you mentioned regarding the safety of vaccination during pregnancy, is this for Moderna specifically or for all mRNA vaccines? It's
for all mRNA vaccines. Yeah, if you go to the CDC website, you can find, it just came out maybe just 2 days ago, but it's all mRNA vaccines. They have assessed the data of women who have received it. So when it was actually authorized, the FDA did not exclude pregnant women and said that it should be a decision made between a woman and her provider. So there have been lots of pregnant women who have been vaccinated and they've been followed, and there was no greater incidence of, say, miscarriage or other adverse outcomes in pregnancy compared to historical data.
And if a booster will be required to cover the different variants of the virus, what do you think will be the advantage for the Moderna and Pfizer vaccines versus the AstraZeneca vaccine?
Well, that's a tricky one, but I mean, I would say that it's much easier to redesign the RNA vaccines just because it's so easy to manufacture them and redesign them. It takes a lot more work to do that with the AstraZeneca type of vaccine, a viral vector vaccine. So it would take longer for them to redesign it. And I know that they've already started clinical trials over a month ago now with Moderna, I know for sure, and I'm sure Pfizer is doing the same thing, where they've taken the redesigned mRNA vaccine based on the South African variant And it's already being tested in clinical trials in phase 1/2 trial. And that data should be available by the summer. And they've been talking about potentially even adding on a boost with a variant design to the phase 3 trial that we're doing. Because, as I mentioned, we still have all the volunteers in our trial, and a lot of them are willing to get boosted. So that remains to be seen. But I just think it's much easier to pivot with the RNA vaccines into a redesign for the variants.
Okay. And is there any status regarding these vaccines in Africa?
Yeah, so a lot of them have been tested already in Africa, particularly in South Africa. So I know for the J&J, AstraZeneca, and I think Pfizer now as well, not Moderna as far as I'm aware, but and then Novavax as well, which is one of the recombinant protein vaccines, which will likely be the next one that will get a EUA here in the US, but all of those had sites in Africa. So there's a lot of data, particularly from South Africa, for several of these vaccines.
Mm-hmm.
And shows similar results.
Great. And can you explain why children are not as likely to get COVID as adults?
No, that's a simple answer. I would say they're less likely to have symptomatic infection compared to adults. Why that is, I don't know. And I haven't been involved in studying that, and I'm not a pediatrician, but I don't really think from what I've seen so far that we really understand why it is.
Mm-hmm. Have you encountered hesitancy to receive the vaccine and how to handle those questions, especially with patients from minority populations?
Sure, and we had hesitancy, for enrolling into the trial even before we knew they worked. So our approach to that was to always be as upfront and honest, and first of all, to acknowledge the hesitancy and a lot of the reasons for it, and not arguing against those, because they're, in a lot of cases, the reasons are real, especially in certain communities of color, where there's been really horrific things done in the name of clinical research before. So to acknowledge that first and foremost, and then to be as transparent as possible about, there are side effects, being upfront of what they are, but to say too that that the data that we have from the clinical trials did include a lot of people from different communities, and that the safety and the efficacy was shown in those groups, as well as the study population overall. So I just think honesty and listening are the two most important things when you're, and not to get angry and not to say you're stupid, and is to try to understand and to reason. That's all. I think that's all we can do.
Yeah, and on that topic, did you do anything specifically to encourage enrollment of minority populations into your study?
Yeah, so we made a concerted effort to have a diverse population in our trial, for the reasons I mentioned before. So we did a lot of outreach, and that was one of the reasons we partnered with School of Public Health. They have existing partnerships with, community groups, across the city in different, wards. So we built upon those existing research networks and relationships. But we also did a lot of media spots, including there's people on this call from Howard. I went onto the Howard radio station a couple of times. I did some interviews with Kojo Nnamdi on NPR, and we just tried to get the word out as much as possible. We had great plans we have a mobile unit that we wanted to take out and enroll and vaccinate people in different parts of the city. Unfortunately, we weren't allowed to do that by the company. They wanted to control the cold chain and so on, that they were worried about us doing that. We were kind of disappointed about that, but that was another way we were originally planning on getting to different parts of the city.
Mm-hmm. Great. Have any research started about which protection is effective, naturally infected or VY vaccine?
So not directly compared, but we do know that after someone gets infected naturally, or in the community, that they are relatively protected for a certain period of time. It looks like, 3 to 6 months at least after that infection. Whether or not the vaccines, the protection is better and lasts longer, we think so, and that's mainly because the antibody levels that we see with at least the RNA vaccines are higher, often a log-fold higher than we see in people who have recently been infected. So there's a lot of scientific reason for thinking that the protection from the vaccines will be better than that induced by vaccination, induced by infection. Right. Yeah. And I think it remains to be seen. We need the time to pass before we can say that the vaccine works for this period of time. That's why I think we don't know, and it's possible to predict really if we're going to need to give boosters and how frequently they'll have to be given, because the time hasn't elapsed long enough for us to be able to tell how long the antibody levels are going to last after vaccination.
Mm-hmm. And did you translate consent forms or recruitment materials into different languages? So
everything was translated into Spanish, but that was it. And we had no control over that really, because of there was 100 sites doing the trial across the country. So the decision was made to try and have everything translated into Spanish, but not other languages.
Okay. And what do you know about so-called inexpensive vaccines that are being developed that would increase availability to third world nations?
Right. So, that's one of the rationales for the AstraZeneca vaccine. They are able to manufacture it quite cheaply. And I know people that I still work with, actually, or collaborate with them. Peter Hotez's group, who used to be based here at GW, is now at Baylor College of Medicine. They have developed a recombinant protein vaccine and licensed it to an Indian company that is now making it. It's currently in clinical trials. That can be made very cheaply as well. And there are other vaccines being developed by Bharat and Serum Institute of India, for example, they can make them quite inexpensively. And that's going to be really important for increasing vaccine coverage in other parts of the world.
Mm-hmm. Great. And did you feel any pressure by the media or expectations to get the vaccine out so quickly?
I would say no, not by the media.
Okay.
I think we put a lot more pressure on ourselves than we got from anyone else, to be perfectly honest. We want to get out of this, pandemic too. So I personally never felt any pressure. We got lots of interest and questions being asked, but I never felt pressure to do things quickly. And just to get back to that, doing things quickly, I hope I explained that although the things were done quickly, they were still done in we did the same number of trials we would otherwise do. They were done in the same design. They enrolled probably more participants than we otherwise would have. And I think too that, people might say, well, now we're seeing these adverse effects, like these rare blood clots and everything. And, even if the trials had been done the regular way, the traditional way, and taken as long as we still wouldn't have picked up those rare events, because they're rare. It wasn't a function of the trials being done quickly that they weren't picked up before. It was the fact that they're rare. So you're only going to see them when millions of people have been vaccinated, and that's only going to happen after licensure or an EUA. So, and the other thing is they're not long-term effects either. Even though they're rare, they're still happening very close to vaccination, which is what you normally see with even rare side effects of vaccination. I've been working on vaccine development for 20 years, and I've never seen a long-term, really serious adverse effect of a vaccine.
Yeah, and on that topic too, so you've worked on many vaccine trials over the course of your career. How has this trial been different than other vaccine trials that you've worked on?
Well, the speed at which we enrolled it. And I had never actually done a phase 3 trial before. I've done lots of phase 1 and 2 trials. So for me, it was a bit new to do a phase 3 trial. And the focus really was on the endpoints. Well, I mean, obviously safety, but also looking at making sure we detected the endpoint. So, you know, looking for, so we had lots of follow-ups with the volunteers to check to see if they had any symptoms, and bringing people in if they did have symptoms that might have been COVID, testing them. So I got a lot of experience, you know, in evaluation of people for potential COVID, and, you know, having to take all the precautions too. If some people, and we did have people who got COVID during the study, and it contributed to those endpoints in the trial. And to, you know, they are saying, so they're getting ready to submit the actual licensure application, the BLA, in the, I think the database lock is on Tuesday next week. And there's been over 900 symptomatic cases now in the trial in those 30,000 people.
Mm-hmm. Interesting, yeah. Do you feel there will be more variants of the virus that will develop in the future?
I mean, most likely, yes.
Mm-hmm.
Viruses, that's what they do. They change. So I think it's likely.
Mm-hmm.
Whether or not the vaccines will, you know, protect against them, that's really hard to say
Mm-hmm.
or predict.
And are you and your team finally getting a chance to slow down and recover from the stress of this rigorous and fast effort into vaccine development, or does the intensity continue as new things appear, such as variants?
It's finally, I would say now, gone back to normal or almost normal in terms of pace. But, you know, we had a lot of other trials we were doing that we're now pivoting to, because we had to like pause enrollment in HIV vaccine trials and other trials we're doing. So we're trying to like catch up with those now. But it's not as crazy as it was like, say, 6 months ago.
Mm-hmm. That's good. Yeah, you definitely all deserve a little bit of a rest after this.
Yeah, for sure.
In developing a vaccine, are side effects on all systems in the body considered in developing it, including ones that would not necessarily be expected to show an effect?
Yes, so we follow everything. So we're not just focusing on one organ system or set of symptoms. You know, for the endpoint, we obviously were focusing on symptoms related to COVID, but, you know, we recorded and reported anything, any kind of adverse health effect that might have been experienced, whether it was related or not. So, you know, everything that happens health-wise to a participant gets reported and recorded.
Mm-hmm. And is there a shortage of raw materials for producing these vaccines?
As far as I know, for the mRNA vaccines, no, not a shortage. I think there have been some hiccups in supply, but I don't think that's significantly impacted on manufacture and delivery of doses.
And in the trial, did any participants have the flu?
Good question, actually. So everyone we brought in for testing for COVID, we also tested them for flu. And no, we didn't have a single person who tested positive for flu.
Interesting. Yeah, social distancing, I think, helped decrease the flu this year.
Masks work.
Mm-hmm. What ethical considerations did you consider when conducting this clinical trial?
Well, all of the usual ones. So, you know, we spend a lot of time in informing and consenting people before they came into the trial, or before they were enrolled in the trial. So, you know, the usual principles of consent and, you know, equity, which is one of the reasons too, we try to enroll as diverse a population as possible, at least reflective of the DC population.
Mm-hmm.
And then, you know, everything went through the usual IRB review process. No steps were skipped. So we followed all of the usual ethical principles and guidelines. Was the IRB process easier now that you're the IRB chair, serving the IRB chair? It's actually because it was such a big study, it went through the IRB. So it went through, so it wasn't the GW IRB that looked at this trial. Yeah. I heard of one case of a person who is of the opinion that she has long COVID post-vaccination. Has this been addressed in follow-ups? Well, it would be followed. We have not had anyone in our 350 people who have anything like that. But yeah, all symptoms and, even ones that might be consistent or similar to post-COVID would be recorded and reported to FDA.
Mm-hmm.
So yeah, definitely that kind of a occurrence would be evaluated and recorded.
Mm-hmm. And someone asked, why is a headache a common side effect of the vaccines?
Well, so it's part of usually a constellation of headache, fatigue, sore muscles, and it's kind of like you feel like you have the flu, really, like illness, and it's really because of the body's reaction to the vaccine. It's like a generalized inflammatory immune response, like massive cytokine release that leads to all of these systemic symptoms. I don't want to go into the pathophysiology of it, but they're all related to just a generalized kind of inflammatory response, because your body's reacting to both the RNA component that's foreign and also the new protein that's being produced. Okay. Is there a reason why the people tend to have more serious side effects after the second shot? Well, it's your body your immune system has already seen it. So it's like a recall. We generally see that with most vaccines. You're more likely to have more of a reaction after the second dose, and because of that recall effect, if you will. Yeah, that makes sense. And for the future, is the parking lot satellite a likely improvement, barrier reduction for recruiting? Oh, yeah. So, yeah, I think that definitely did help with recruiting. Unfortunately, that's was just lent to us. And we've been receiving, I think they want it back, to be honest. So we've been having to justify why we still need it there. So unfortunately, it won't be a permanent addition to the GW clinical trial effort. Yeah. Well, great. With that, I think we can wrap up. There's no more questions. But thank you so much for this presentation. It was a great way to start out this conference. So next up, I'll introduce Mariel Hayes, who will be moderating the next panel. Mariel, I'll turn it over to you. Thank you.